Hello everyone, I hope you all are doing well. Today in this video I am going to share my IISc Bangalore interview experience for MSc Life Science program. Now there was 2 to 3 panels and almost 5 to 6 professors were there in each panel and they are very friendly. Now when I arrived after IT verification they called me for interview. In the interview call later they won't specify the particular interview time. They only say you the name of the slot either in the morning slot or in the afternoon, either from 9 a.m. onwards or from 2 p.m. onwards. And when you reach there after ID verification, when your time comes, they will call you for the interviews. Now, um, I arrived around 9 a.m. and I have to wait for 1 hour or so. And after that, they called my name. When I entered the room, they were very friendly and they asked me if I had my meal. I told that yeah, I had, but it was in the early morning, and that was around 11:30 almost the time when I entered the enter for interviews. Then she asked, early morning? Oh, so what is the early morning for you? Exactly what time? I told that I had my breakfast around 6:30 in the morning. And then they said that, oh, then you must be very hungry right now. But you have to manage your hunger for 15 to 20 more minutes. Then after that, we will bring some food for you. And then I said, okay, okay. Uh, After that, they asked me my academic background from where I am pursuing my BSc and uh, with what topic I am most comfortable with, from where they can ask me questions. I told that I like cell signaling, transcription factor biology. Then one professor was there. He asked that can you name a membrane protein, and a protein found in the nucleus? I told that yes, like many receptors, receptor tyrosine kinase or GPCR, they found in the cell membrane and inside the nucleus. And there are proteins like nuclear lamina, and that is made up of laminin proteins and our genetic material that also contain protein. Then he asked, our genetic material is DNA. What is the protein? I told that during the packaging of DNA, they wrap around the histone proteins. Therefore, during packaging, there is also protein is involved in along with DNA. Uh, then he asked me if you have to detect a membrane protein and a nuclear protein, how would you detect? Uh, I told that for membrane protein, for detection of the membrane protein, I would design antibody against the protein and add fluorescence stack with the antibody. And after the antibody binds to the membrane protein, I would get a signal under fluorescence microscope. Then he asked, okay, uh, then how would you detect a nuclear protein? Antibody are very large and they cannot really penetrate the cell membrane. So how you would allow the antibody to penetrate the cell membrane? And I was thinking, what modification I have to do in the antibody molecule. And after some time, I told that like the way we transfer genetic material like DNA inside a cell, physical methods like micro injection or particle bombardment, we can use such techniques, and by this way I can directly transfer the uh, antibody into the cytoplasm. Then he told me that these techniques are mostly used for genetic material, but uh, not for proteins, right? I said, yeah, but we can use it for protein also. Then he told me, so the antibody has entered inside the cell, has entered inside the plasma membrane, but how would you modify it to enter the nucleus? I told that uh, for for allowing it to enter inside the nucleus, a protein must have nuclear localization signal, and If a protein have nuclear localization signal, that will be recognized by important molecules which are responsible for transporting a protein inside the nucleus, and we have to add a nuclear localization signal in the antibody, and then it will bind to the protein and it will move inside the nucleus. Then they said okay. Then there was another faculty who asked me questions. He told me if I was comfortable with biochemistry. I said yes, you can ask me questions. Then he told me to draw the Ramachandran plot. There was a whiteboard and a marker. I draw the Ramachandran plot. First of all, I forgot which axis was psi and which was phi. I was confused. Uh, then I wrote the name psi and phi. After that, he told me to draw the location of the alpha helix and beta sheet, where right-handed alpha helix and where will be beta sheet. I draw their locations, and then he asked, what is the most sterically hindered amino acid? I said proline, and glycine was the most relaxed. Therefore, glycine will occupy the most of the space in the curve. Then he asked me, uh, if I have a protein, how would I experimentally determine if it is alpha helix or beta sheet? I told that CD spectroscopy is mostly used for determining if a protein is alpha helix or beta sheet or random coil. Then he asked me, like, he told me to tell name of one right-handed alpha helix protein and another left-handed alpha helix protein. I told that left-handed alpha helix are not that much abundant inside our body because the structures are not uh, very stable structure. Right-handed alpha helix are mostly found inside the body, and I told like the receptors which are alpha helical structure like GPCR which contain seven alpha helical structures. This is a right-handed alpha helical structure, but I could not remember the name of a left-handed alpha helical structure. Then he told that you said cell signaling is your topic of interest. So can you say something about calcium signaling? No, no. Before that, he told me where calcium is more abundant, outside the cell or inside the cell. I told that outside the cell there is generally more calcium, and in response to certain signaling pathways, calcium, the amount of calcium increases inside the cell. Like calciums are mostly stored inside the endoplasmic reticulum, and in case of IP3 signaling, the IP3 gated receptors are activated because IP3 bind to the receptors and calciums are released from the endoplasmic reticulum. And when the signaling is uh, over, the reuptake of the calcium ions occur inside the endoplasmic reticulum. Then he asked how the reuptake occurs. I told that the release is generally a passive process which occurs by binding of inositol triphosphate uh, with the IP3 receptors, but uptake is an active process which requires binding of ATP molecules, and by this way uptake occurs. Then he told me, can you tell me any other situations where calcium signaling is also involved? I told that yeah, during muscle contraction, uh, calcium ion is also involved because there are accessory proteins like troponin, tropomyosin, and troponin has calcium binding site. During muscle contraction, calcium binds to a troponin molecule, which ultimately leads to the contraction of muscle. And then he asked, "Can you tell the event which occurs during muscle contraction?" And I told that yeah, during muscle contraction there is a motor end plate where the signal from the neuron ultimately which comes from the brain and it reaches the motor end plate, and in the motor end plate uh, there is acetylcholine receptor uh, in the postsynaptic neuron where acetylcholine binds to the acetylcholine receptor, and upon this binding calcium uptake occurs, and uh, as calcium enters this causes depolarization, and T tubule, calcium comes up from the T tubules, and ATP molecules are also involved in the muscle contraction, the power stroke, that things I told them. Then they said okay, and these were the broadly the questions which they asked me. And after my interview is over, as they have told me, they gave me some food to eat, and I took the food and went to another room where they have offered me tea or coffee. And this was the interview experience. They were very friendly when I stuck into some point. They try to give me hints such that I can proceed with my answer. And they generally don't try to humiliate you, or you don't know the things, or laugh at you. Sometimes they even try to give you hints, or they in fact say the answer sometimes. This was my overall experience for IISc Bangalore interview experience. The result is yet to come, and I recently qualified the second round of interviews for TFR Mumbai, and I will make a separate video for my TFR Mumbai second round interviews. And for integrated PhD at IISc Bangalore, the interview is on 19th May, and I will again visit Bangalore for my interviews. Actually, there are three interviews on the three consecutive days on 19th. On 19th, there is IISc Bangalore interview for integrated PhD. On 20th, there is NCBS interview for integrated PhD, and 21st, there is interview at JNCSR for integrated PhD in biological sciences. Therefore, once I visit Bangalore, I would complete all the three interviews and return back to Calcutta. I would make a separate video for those interviews. Okay. Keep reading, keep studying, stay motivated. All the best. Bye bye.